very much for the invite. Certainly in putting this talk together, I think I learnt a lot actually, because I think when you can't hear me? Mike?
Yes.
I can.
Hold it up.
Oh, sorry, I'm a very quietly spoken person. No, I'm happy. So I'm very much a patient-facing clinician, but I think whenever you're talking about genomics, it's such a large subject. So I feel that this short 20-minute talk needs to reflect genomics worldwide, really. So I'm covering 3 topics, and I'm kind of doing it backwards, actually. So I'm going to tell you a little bit about clinical care and just explain what genomics is. I have here colleagues in the audience, including Mina herself, so some people are very knowledgeable about genomics, and I'm certain that some of you will have read articles about it, or maybe you are clinicians who offer genetic testing to your patients, etc. But I am going to tell you a little bit about references and data sets, which sounds a little bit dull, but actually forms the foundation of how we approach and offer patients our genomic tests as well. And I will also touch a little bit on research. Everything that we do in clinical genetics is based somewhere in the large amount of research that people have done over many, many decades. I'm not a researcher myself, but I'm there to support my patients if they are interested in entering research studies. So these are the 3 main sort of areas I'm going to cover. So starting a little bit with what is genomics. So genomics is the study of an organism's genome, so all of their genes. And the aim is to understand those genes on an individual basis, but how do those genes interact with each other, and how do they interact with the environment as well? A little bit of science. I'm sure many of you will know some of what I'm going to say, but we're all made up of trillions of cells, and inside the center of every cell we have a set of our DNA. Okay, it's made up of lots of chromosomes, 23 pairs of chromosomes, and lined up along those chromosomes are our genes. We have roughly 20,000 or so genes, and when it comes to trying to understand what those genes do, I'd probably say we still don't understand what the vast majority of those genes do. We probably understand about 6,000 to 8,000 of them in terms of their how they cause human disease, but even for very well-known genetic diseases, there's still an awful lot to learn. In terms of how we test genes and DNA, I'm not going to probably go into too much of that in this talk, but to say very broadly, we can look at things on a chromosome level. So do anybody have extra bits of chromosomes or missing bits of chromosomes? But then we also talk about DNA sequencing. So people talk very much about reading people's genetic code. So what they mean is reading these little chemical letters to try and understand whether there is a small alteration somewhere in that gene. When it comes to explaining what genomics is and how it's used, it is used across many different facets of healthcare. My role in clinical genetics is to see individuals who have what we call rare disease. So I've got 3 very quick examples here. So at the top is a little boy who has Down syndrome. This is what we call a chromosomal condition where they is born with an extra copy of chromosome 21. The picture of Angelina Jolie, obviously, she was found to have an inherited form of breast and ovarian cancer in her family, and she had genetic testing for that, and then therefore she had appropriate clinical management. And then we also in clinical genetics see people who've got very, very rare syndrome. So KBG syndrome, many of you may never have heard of it, and actually when we see families, sometimes the diagnosis that their child is given is a completely unknown diagnosis to them. They meet lots of people and clinicians and doctors who have never heard of that condition. So patients, families become very much the educators for healthcare professionals themselves, and sometimes they find that very frustrating actually. So the other sides of genomics and how it can serve patients is in the realm of personalised medicine. So personalised medicine is where you want to be offering treatment to patients based on who they are. So you're not saying that everybody with breast cancer necessarily gets the same treatment, or everybody from a particular ethnicity necessarily gets the same treatment, but it's much more tailored towards that individual person. So this can be within infectious disease if you are sequencing the pathogen itself. It can be in trying to understand how people may react to certain drugs so that you can give them the appropriate dose, or maybe not give them a particular drug at all. And then when it comes to tumours, these are not necessarily inherited tumours I'm talking about, which is what we tend to see in clinical genetics, but actually across many different types of cancers, people are offered a type of genetic testing to actually look at the genetic makeup of that tumour to see how best you can actually treat and manage it. So I'll just tell you a little bit about rare disease because that's what I tend to see more in my job. Many people think that the word rare means that there's hardly any people around who have a rare disease, and actually what do you do in clinical genetics? You must have hardly anybody to see, and that's actually not entirely true. 1 in 17 people in the UK will develop a rare disease, so that equates to roughly 3.5 million people in the UK. And the definition of a rare disease is where that individual condition affects less than 1 in 2,000 people. In genetics, in my job, we see people with ultra-rare conditions, and these are conditions which affect less than 1 in 50,000 people. 80% of rare disease is genetic in nature, so that we do try to explain to people that rare doesn't necessarily mean genetic, so there's still a sizable number of conditions which will have some influence from genes, but which are not necessarily seen as being purely inherited. Lots of rare genetic diseases are seen in children, but actually even if you have inherited a particular condition from one of your parents, it doesn't necessarily mean you will manifest that condition in childhood. You could live quite a lot of your childhood and adult life without ever having that condition ever develop. So actually that's another problem when it comes to people accessing healthcare, when it comes to genetics, is people assume you need to have something which affects you right from the day that you are born, but also that you need to have a family history. So we see lots of people who have not been referred to our service because people assume that you need to have multiple people in your family who've got the condition, otherwise it's not genetic. So this is just an infographic which I quite like to show my students because we have what's called Rare Disease Week for Cardiff medical students, where we try to educate them about rare disease and the fact that it's not that rare. So this is just to try to show you that patients who have a rare disease will go through multiple rounds of referral. They will get seen by many different people who will potentially tell them they will maybe manage their symptom in that particular area. So for example, a heart problem, but they may miss the bigger picture of the fact that people have got symptoms in lots and lots of different areas of the body. They may have lots of invasive or non-invasive investigations. They may come across patient support organisations, but one of the problems with not having a diagnosis is that there isn't necessarily an organisation you can go to. If they do happen to get to have the chance to actually have a genomic test, it's a question of have they been offered the correct test, has it been interpreted correctly, and even if they haven't received a diagnosis from a genetic test, there is also the problem of people being told, well, it's not genetic. So part of our role in clinical genetics is not to necessarily offer people tests, but to educate them as much as possible on the test that we're offering or the diagnosis that they have, so that they can make decisions as to whether they want to proceed or not. And to say to them that even if I don't find a cause on a test and I can't give you a report that has a name on it for a condition, it doesn't necessarily mean it's not genetic. So I think a lot of what people have already talked about in previous speakers, a lot of it is also relevant for genomics and genetics as well. So in order for people to access genetic services or genomic services, somebody needs to recognise that that person may have a condition which is genetic in nature. And that might not be the health professional they're seeing. I've met patients who have diagnosed themselves, sometimes with ultra-rare conditions, because they have gone onto the internet, they've done lots and lots of research, they've put all their symptoms into Google, and they've eventually found something, and they've done all the reading. And then they go to their GP or they go to another health professional and they say, this is what I think I've got. And then the doctor themselves has never heard of it, but they will eventually, hopefully, get a referral to us. One of the other problems is people don't always know that we exist. I think that's changing. I think because genetics is very much out there in the media space, I think lots of people will just ring us or call us or tell their GP to refer them or somebody else to refer them. But I still see patients who say, we didn't know where to go, my doctor didn't know where to send me. So that is still an issue. The last one, I think, is probably true for any healthcare service. Your patient needs to feel that they can trust the services that they're coming to. I'm going to talk a little bit more about that when it comes to research. I think research in genetics has sometimes had a little bit of a shady past, to be honest, and people don't always trust that their data, when it comes to genetics, is going to be used in an ethical way. So education, as I think in clinical genetics we do quite a bit of education. Our role is also to try to help and support our non-genetic specialist colleagues so that they can offer genetic testing to their patients and explain it in the best way possible. So this is just some examples of websites that are out there. Some of them are for clinicians and for other healthcare professionals as well, like specialist nurses, for example. But there's also lots of patient websites as well. So Unique, which is down here at the bottom, is a website I use a lot. It provides booklets that have been curated by families. But then there are also people who've never managed to receive a diagnosis for themselves or for their child, and that's when it gets difficult because there isn't that group that they can go to. So Swan UK is a well-known charity where people can go to get support even if their child hasn't got a specific diagnosis. In terms of education, we regularly get asked to go to different specialties and try to help them in understanding genomics because it is getting very, very complex. It's such a large area that it's very difficult for us all to keep up to date. There are hundreds of genetics journals. There are thousands of studies across the world. Every time I go to a meeting, there's some brand new study or something like that. It's actually very, very hard to keep up with as a subject. I sometimes see our job sometimes as being a bit like a GP in the sense that we can see anything and everything. We see any age group. We will see families. It's just that we see the very rare side of things as opposed to people in primary care. So I'm going to move on to a little bit about references and data sets. And this is just to try to explain the fact that even if I see a patient in clinic and I'm offering them the best test, even if I've done the best I can, there are some inherent issues with inequality when it comes to the reference information that is out there on which the test is based. Okay, I've got an example here of a patient who's had their genome sequenced, and I've only illustrated what I'd say is 30 base pairs, but we've actually got 3 billion, and obviously the slide's not big enough. And then below that is the reference DNA. So in the same way that if you went to your GP or any doctor and you had a blood test, so for example you're getting your hemoglobin checked, they're going to be comparing your reading against a reference range, and that reference range has been created over many decades because they've sampled thousands of people from all across the world, and everybody has agreed that this is the normal reference range. So when we're doing genomic testing, patients' DNA is getting compared against a reference. So the big question is, who is the reference? Okay. So a little bit of evolutionary history. So you'll all know that our nearest relative are chimpanzees, and they say that if you were to sequence a chimp's DNA and you sequenced our DNA, that it would be 98.8% similar, which sounds really pretty similar, but obviously there's some large differences between humans and chimps. So that 1.2% difference, which doesn't sound like much, is actually the same as 35 million different DNA sequence changes. So I showed you on the previous slide one little base pair change. You can imagine 35 million of those is what makes the difference between one of us and a chimpanzee. So if I picked any two people in this room and sequenced your whole genome, which I can't do myself, but I would ask someone in our lab to do that, there would be roughly 1 in every 1,000 base pairs would be different between any two people. And that doesn't necessarily mean you have to be from a different ethnicity. You can literally be from the same village and you would still have a significant number of differences between those two people. So that equates to about 3 million differences, which is 0.1% of the total genome. It's not very much, but it's still enough to account for why we all look different and why we've got different sized hands and different shaped faces and all of that. So apologies, I'm not very good with this mic, it doesn't like me. So for every single one of those differences, we can't assume that they're necessarily the cause of human disease, and that's something we have to explain to our patients, that it's not enough to find an alteration in a gene. It has to be the right gene. We have to have enough understanding to be able to then finally say to you, yes, we believe this is the cause of your symptoms. So for each difference, it may have no effect on that gene whatsoever, or it may have some slight effect on that gene, which accounts for the diversity that I can see in this room when I look at all of you and the diversity you see within the human population. That's what's so wonderful about humans. We're not all exactly the same. But what we're probably interested in when it comes from a medical perspective is those little changes which are having some adverse effect on our health. But we're also very interested in those that are having a protective effect as well. So to tell you about the reference. So the reference originally came from the Human Genome Project. So this was a very, very large collaborative project. It took place over many years, many billions of dollars from across different countries. And all they wanted to do was just get one reference, one nice long stretch of 3 billion base pairs, which was to say this is the human genome. And it's quite hard to actually find out exactly who the reference was, but I think it's a small number of Caucasian individuals. And one paper I read said that 70% of the human genome was just from one person. Okay. Now that reference has been updated because the Human Genome Project was completed 20 years ago, and now the current reference, or what is the agreed reference, is updated on a regular basis, and we're on version 38. So every time someone has a genetic test, their sequence is being compared against this current reference. Now that reference is much better than it was. It includes more people from across the world, but it's still very much based on people who are of European descent. And that's one of the biggest problems with the reference, is that it doesn't represent human diversity. And therefore, if you're going to be compared against somebody who is not of your ancestry, then there is always the problem that you're either going to miss something that your test should have detected, or you're going to incorrectly say that the particular change that's been found is disease causing. So there's problems at both ends. So it's definitely not a gold standard genome. It's not a perfect human being, this reference, and certainly it needs a lot more work. And this is just one example of an article that the BBC covered sort of last year talking about missing genomes. So going to the middle bit, which is all about research. So I don't think this is just in relation to genomics, this is in relation to research across health, that it's historically been limited due to structural racism, language barriers, economic challenges and socio-demographic features. But when you talk about genetics, then there's obviously this problem of legacy of racism and misconduct within genetics. There are certain populations who will completely shy away from having any involvement in genetics research because of history. And that is going to account for some of why certain populations are not that keen to include themselves. That therefore means that those who are offering research need to be able to explain it in a culturally sensitive way, to build trust with communities in order to help understand why those barriers are there and how they can help to lessen those barriers. There's obviously a lack of access to computer-based resources. We've all got phones, we've all got computers, and especially in the West, we probably take it for granted that we've all got those things. But there are lots of people who don't have those things. And even if they do, if the resources that are on there about a particular research study are not in their language, or even if they are in their language, they're in complex genomics language, then people are not going to understand what the research is about. Genetics is a language that's completely new. When I entered the specialty, it's not the same as any other healthcare profession. You have to learn lots of new words and ways to explain those very complex concepts to people from every educational background. So this is a big problem with research in general. Those with disabilities are often excluded from research. They may only be included in research if it's about that specific disability. And even when they are included, there can be the feeling that that research's aim is actually to eliminate that disability. So that, again, is another problem as to why people don't always come forward. And then obviously there's the lack of culturally tailored counselling. That's not just within explaining research projects, but within clinical genetics care as well. And different centres around the UK, particularly Bradford Clinical Genetics Service has very much led when it comes to trying to offer culturally tailored counselling for their patients and making sure that they have enough translators and that they have leaflets and are able to explain things in the most sensitive way. So I'm just giving you two examples of very large research studies that are being done in different parts of the world. Many of you may have heard of the 100,000 Genomes Project. That's been going around for a long time now, but that's a UK-based project. But actually there are some very, very large projects that are happening all over the world because The cost of doing a genome now is down to a few £1,000s. So this is now technology that is not affordable to every nation, but is becoming more affordable. So Genome Asia, I couldn't find a figure for how many genomes they've managed to get, but they certainly are looking at, I think, over 64 countries. And H3 Africa is also a study that's been done by the African Society for Human Genetics, and they've also had some support from the UK and the US as well. Yeah. So it's a UK-centred document, but it echoes exactly what's written in the other paper next to it, which is from the US. And I think the talk, what I've given so far, has given bits of solutions as well. So just to kind of summarise what people are saying out there as to how do we address some of the inequality, or start to address the inequality, is genomic literacy. So I've already talked about education of healthcare professionals, but education also of the public. So there are initiatives out there that go out to primary schools and secondary schools to try to help educate them about genetics and genomics. And it also helps to diversify the workforce, because if you have more voices from different backgrounds, then you are more likely to, that's likely to then reflect in the way that you offer services to patients as well. So community engagement, lots of the projects which are ongoing where they are trying to approach people from ethnic minorities, they first of all have to go out and just talk to them. You can't start with research and start giving out leaflets and saying to people, hey, give me your blood sample. You actually have to go out there and talk to people, see how they feel about it, ask them what's best for them, so that you can then make the resources and build that trust with people out there to make the research more successful in the long term. The research needs to be not just on genetics, so it doesn't need to all be about blood samples and reading genes. It also needs to be about understanding people, understanding the barriers to research participation, and understanding the social, legal, and ethical implications of genomic technology. Not everybody sees genomics as a positive thing. It, I think, gets shown that way in the media, that it's going to solve everybody's problems. That's not necessarily the most accurate way to represent genomics, and not everybody feels that way. A lot of people are very worried about having a genetics test. They're worried about how people will see them. They're worried about telling their family. So these are all conversations that need to be had to understand how people feel about genomics, and I think that's going to be ongoing for many, many decades to come. And I've already touched on increasing diversity in research databases. That was my last point. So I just thought I'd end with one small UK study. This is being done by Bradford and London. Jointly, they're trying to sequence or look at the genes of 100,000 people. They're trying to understand, particularly in Bangladeshi and Pakistani people of that origin, to try to understand their health in relation to genetics. So there are small projects. They're not necessarily as widely publicised as 100,000 genomes, but certainly lots of people are trying to start working on inequity in genomics. Thank you very much.